Okay, are there any questions?
I have a quick question. It's not lab related though.
Okay.
For my question that I answered for the lecture worksheet, it was how does the size of eukaryotes compare to those of prokaryotes? And I did a mathematical difference between the diameters that were given to us in the book. But then in the lecture earlier, you said it was 10 times, where it came out to like 50 times mathematically, like on average. Will either answer work, or would you rather that say 10 times so that everybody's on the same page?
Did you say that it is 50 times larger, or did you give a mathematical answer that one cell is such and such and the other cell is such and such?
So I said that eukaryotic cells are typically 10 to 100 micrometers in diameter as compared to prokaryotics 0.2 to 2 micrometers, which mathematically comes out to 50 times large.
Okay, yeah, that your answer is fine.
It is? Okay, thank you.
Yeah. All right, any other questions before we begin? If not, let's share the screen, I think. Well, let's begin. We don't need that page. I'm not going to close it now because it might shut down the converting of the lecture that's going on. There you go. You can see convert meeting recording. It's only 43%, which is actually pretty good because I just stopped it about 20 minutes ago. So that'll interrupt, and sorry about that, but it used to finish before I started the lecture, but that has been a while. There's a syllabus. We're going to be doing lab 6 today on disc diffusion and antibiotics. And I need to tell you to read the unknown project instructions. You can find it on Canvas. You need to read that for next week because I'm going to talk about the unknown project. And we'll start the unknown project next week. Let me think about that. Do I want to do lab 7 on Thursday and talk about the project on Tuesday? That will probably be best, and that way you can work on the unknown project on both Tuesday and Thursday. And then we'll have something to do on Tuesday and Thursday. So that's probably what we'll do. In the future, we'll only have lab 8 and unknown work on both Tuesday, and then on Thursday, we'll only have unknown work. So there really won't be a lab on Thursday next week. But I think this coming time, this next week, we will probably do this on Tuesday and then lab 7 on Thursday. All right, any question about the schedule or about what to do? If not, I'm gonna shut that down because I've got a lot of windows open.
I had a quick question.
Uh-huh.
Are we gonna finish talking about eukaryotes next Tuesday from the slide that we were just going over?
We just shut down the syllabus, so.
Because it looks like next week we're supposed to do chapter 5.
Let me take a look at that. I don't remember off the top of my head, so.
If not, I was gonna. Okay.
Let me reopen up the syllabus. Look at that. Yeah, next week we are supposed to start chapter 5, so we will talk a little bit about chapter 4, but on the eukaryote organelles, I'm going to be skipping all of them because you supposedly covered eukaryotes and organelles in your preliminary class, Biology 160. And okay. I will talk about the endosymbiotic theory in the lesson, and that's like about 3 or 4 slides. But everything else I'm going to skip, and I'm going to assign it, so I will make those notes available to you. But You should have already had that material, so it's supplementary material.
Yeah, purpose of my question was just wondering if we could get a copy of the PowerPoint.
Yeah, as soon as I finish with the lesson.
Perfect, thank you.
All right.
And I have a question. Are we getting tested on chapter 4 next week, next Tuesday?
On Tuesday night, Wednesday, assuming the schedule is correct, then you guys, you have to tell me in advance when something isn't correct, and then I can switch it. I didn't know until afterwards, and people had already started or taken the exam, so I wasn't going to change it. But I remember quiz 2, I think it was quiz 2B. It didn't start at 8 p.m. like it was supposed to. It was starting at midnight. And like I said, if you guys let me know in advance, I can correct that. I didn't correct it because I didn't find out until afterwards. So try and notify me in advance. And if you can tell me rather than email me, somebody did email me after the lab, but I didn't check my email until the next day. So it was way late before I found out about it.
I actually emailed you because I was trying to take that test that same day.
Yeah, you should have talked to me in the lab. Like I said, if people let me know in advance, I could change it. I wasn't going to change it after people had already taken the exam, and I'll go in there and change it. What happened was, it was only last term that students asked for a different time frame to take the exam. And they took quiz 1 and quiz 2 on from midnight Tuesday night to midnight Wednesday night. And somebody was working, and so they asked to take it from 8 p.m. to 8 p.m. And then we discussed it, but this was after quiz 1 and quiz 2. And so I changed it. So all the others should be correct. I thought I went back and changed it, but apparently I missed quiz 2B. And that was my mistake. But if you see something like that, notify me in advance and then I can correct it.
So is the timing going to be for future quizzes from 8 p.m. until the next day?
And they should be from 8 p.m. on Tuesday night to 8 p.m. on Wednesday night. Okay?
Thank you.
However, if there's a mistake, just let me know. All right, let's go to lab 6.
Can I bring up one more thing real quick?
Yeah.
So quiz 3, it's showing on the syllabus for this coming week, but in Canvas it shows for May 11th.
Well, obviously that's a mistake. Can you notify me after the lab and I will change that?
Yes.
Okay? All right. Because I'm going to forget by the time I finish the lab. All right, let's go back to the lab. This is the worksheet. Here we go. Lab 6, disk diffusion, the evaluation of disinfectants and antiseptics, and the effectiveness of antibiotics. In this lab, we're mainly going to be looking at disk diffusion using antibiotic effectiveness, but we can use the same procedure to look at the effectiveness of disinfectants and antiseptics. So do the reading before doing the lab. There are supplemental materials. You're going to be watching some video clips and performing a virtual lab. Upon successful completion of the lab, you should be able to identify the type of chemical agents and the active ingredient used in various disinfectants and antiseptics. 2, evaluate the effectiveness of the disinfectant and an antiseptic using disk diffusion. 3, determine the mode of action of various antibiotics. 4, evaluate the effectiveness of various antibiotics using disk diffusion. And number 4 is what we're mainly going to look at in this lab, looking at the effectiveness of various antibiotics using disk diffusion. but also lists the variables that influence antibiotic disc diffusion susceptibility testing. And there will be at least one question on that. Be able to define the terms, what's sterilization, disinfection, disinfectant, and what's an antiseptic, a bactericidal agent, a bacteriostatic agent, what's an antibiotic, what's the spread plate method, and then what is a zone of inhibition. And I usually abbreviate that ZOI, zone of inhibition. So when evaluating disinfectants and antiseptics, you should know that sterilization is the complete destruction or elimination of all viable organisms, including endospores. Sterilization may not remove, what do you call it? Prions, okay? It is very difficult to totally destroy prions. And so usually when we sterilize something, which will kill an endospore, it may not kill a prion, which I don't know whether a prion is alive or not, but it will not eliminate the prion, okay? Disinfection is the destruction of vegetative cells, not necessarily destroying the endospores. Like when we disinfect a toilet, we will kill vegetative bacteria cells. But if the cell has made an endospore, we won't kill the endospore. Any question about that? So that's the difference between sterilization and disinfection. Sterilization kills endospores and essentially all cells. Disinfection only kills vegetative cells. Disinfectants tend to be chemical agents, and you apply it to an inanimate object. An antiseptic is also a chemical agent, but it's applied to a living tissue. For example, if I were to use bleach on a toilet, I would call it a disinfectant. However, if they use bleach on a patient, and they used to use this for necrotizing fasciitis, that's the flesh-eating bacteria, we would say the bleach is an antiseptic. Any question about that? All right. To evaluate the effectiveness of chemical agents, the surface of a nutrient agar plate is usually inoculated with a particular bacterium. Paper bacteria discs saturated with different chemical agents are then applied to the agar surface. The chemical agent diffuses into the agar medium, establishing a concentration gradient around the paper disc. Sorry about that, let me save that. So the chemical agent diffuses out of the paper disc. We then incubate the plate overnight and then look at the bacterial growth pattern. If there's a zone of inhibition area around the paper disc, an area of no bacterial growth, the chemical agent can be assumed to be effective for that particular bacterium. Conversely, if there's no zone of inhibition, the chemical agent can be assumed to be ineffective. Now, it's not quite as simple as that, but for this purpose of just talking about the zone of inhibition, if you see a zone of inhibition, you can assume that the chemical agent is at least partly effective, okay? And if there's no zone of inhibition, the chemical is not effective. All right? And we'll talk about fully effective and not significantly effective a little bit later. Here are some common disinfectants and antiseptics, and it breaks them into different categories. Like most people know, ethanol and isopropanol, which is rubbing alcohol, are alcohols. And then the table gives you their general action and their preferred use. And alcohols can be used as an antiseptic, but they're really weak antiseptics. So we call them degerming agents, meaning you can apply alcohol to your skin, but it will not kill off all of the bacteria in the area you apply. Okay. Then again, other, antiseptics, which are stronger, like formaldehyde, formalin, glutaraldehyde, or chlorine, like chlorine bleach, we use that as a disinfectant. And they will kill all vegetative cells. They may not kill endospores. Iodine is not considered a disinfectant. We don't use it to clean the toilet, but like tincture of iodine, you used to put on patient's skin, and we used it as an antiseptic. It's not used as much in the hospital. It used to be used all the time before surgery, but now they have other agents that they use. It can still be used, and you can use tincture of iodine when you put iodine on a wound. You have that usually in people's home, and that we use as an antiseptic or a, antiseptic. Heavy metals have come back in their use, like silver is put on dressing to work as an antiseptic on a patient's dressing, meaning a wound cover. They can be used as a disinfectant. Hydrogen peroxide or peroxygen. This one we'll talk about a little bit later in the lab. Hydrogen peroxide and other peroxygens are an antiseptic and a disinfectant, which can be used as an antiseptic. And what's different about these peroxygens is they are an antiseptic that actually can kill an endospore. Most antiseptics do not kill endospores, but endospores are really sensitive to oxidation, meaning that the endospore is usually an anaerobe. And so they're sensitive to oxygen. And so peroxygens can destroy endospores. Okay. All right. Surface active agents include disinfectants. They denature proteins and disrupt plasma membrane. An example of that is quaternary ammonium compounds, which I think that's something that's now used in hospitals. Somebody can speak up if you know specifically. All right, when we look at a zone of inhibition around a paper disc containing a chemical, you should realize that the size of the zone of inhibition depends only partially upon the sensitivity of the organism to the disinfectant or antiseptic. There are various other factors that affect the size of the zone of inhibition, including the concentration of the chemical, the molecular weight of the agent, and the density of the bacterial inoculum. Generally, the more concentrated the chemical, the larger the zone of inhibition. The smaller the molecular weight of the agent, the larger the zone of inhibition. The more dense the bacterial inoculum, the smaller the zone of inhibition. So there are other factors that come into play to determine the size of the zone of inhibition, other than an organism's particular sensitivity to that agent. Let's talk about the evaluation of antibiotic effectiveness. Antibiotics can be categorized as bactericidal if they actually kill the bacteria. So if they kill the cell, then it's bactericidal. However, if the antibiotic simply inhibits the growth of the bacteria, we call it bacteriostatic. How you can separate the two is you apply the antibiotic and then you stop giving the antibiotic. If the bacteria come back, the bacteria the antibiotics was bacteriostatic. It stopped the bacteria from growing, but it did not kill the cells. If on the other hand, you apply the antibiotic and then you stop applying the antibiotic, if the bacteria do not continue growing, then the antibiotic was bactericidal. Now, this is best used when we're talking about applying an antibiotic to a Petri dish, because when you're applying an antibiotic to a patient, there's a couple of other factors that may give rise to whether the bacteria come back, such as, did you apply enough antibiotic for long enough to kill all of the cells, because the cells are not killed instantaneously all at once. Any question about any of that? All right, when a bacteriostatic antibiotic is used, we're relying on the host immune system to assist in the elimination of the infection. So although the bacteriostatic may not kill the bacteria, it stops them from growing, and then the host immune system eliminates the infection. All right. The Kirby-Bayer dysdiffusion test, sometimes called the KB dysdiffusion test, is the most common method used for testing antibiotics. The results of such testing help physicians in choosing which antibiotic to use when treating a sick patient. The bacteria is isolated and then put on a on a Petri dish. So you apply the bacteria everywhere so that we get the bacteria growing everywhere. And that we call a lawn of bacteria. Let me show you that. So other than the zones of inhibition, you can see the bacteria growing everywhere on the plate. It's most visible on the left side because the bacteria is the off-white and the agar is darker. It is visible here, but because of the lighting, it's not quite as visible. We'll talk about how the bacteria is put on in a little bit. We then apply paper discs containing a chemical, in this case, an antibiotic. The antibiotic then diffuses out of the paper disc. We then allow the plate to grow the bacteria, meaning we put the plate in the incubator upside down and incubate it for a certain amount of time. If you're doing the Kirby-Bayer test correctly, you incubate it for 18 hours at 35 degrees Celsius, and then look at the plate, looking for zones of inhibition. Where there is no or very little zone of inhibition, like here and there, we can assume that the antibiotic was not effective in treating this bacteria. If you see a zone of inhibition, then the antibiotic is at least somewhat effective, but the question is it really effective? And to determine whether it's really effective, you have to measure the diameter of the zone of inhibition and then look up the diameter in the chart. And generally, a large zone of inhibition, the antibiotic will be considered effective against that bacteria, but a smaller zone of inhibition may not be effective. And we'll talk a little bit more about how you determine whether the antibiotic is successful in inhibiting the bacterial growth. Any question about any of that? So we don't know on this plate, is this bacterial agent on that disc bacteriostatic or bactericidal, because all we know is the bacteria have been stopped in growing in this region where we have the zone of inhibition. All right, the inhibition zone size is dependent on several factors. It is dependent on whether the bacteria is sensitive to that antibiotic, but it's also on the growth rate of the bacterium, on the diffusion rate of the antibiotic, and the number of bacterial cells that we inoculated on the plate. Okay, so table 6.2 lists the procedural protocol required to standardize some of these variables when performing the Kirby-Bayer distaffusion antibiotic test. You always inoculate nutrient broth from several bacterial colonies grown on non-selective media, and you grow them for no more than 24 hours in a nutrient broth, meaning a liquid. You then adjust the broth turbidity to 0.5 McFarland units, and that's just measuring the bacteria density. This is approximately 1 × 10⁵ organisms per mil, and you only use this amount of cells to put on the plates. You then inoculate at room temperature Mueller-Hinton agar plates. And we only use Mueller-Hinton agar because other media can have a different size of zone of inhibition. The agar is always between pH 7.2 and 7.4. The depth is always a certain depth. And in this chart, it says 4 millimeters. I think this is actually wrong. I think it's always 5 millimeters, but we'll just say it's always a certain depth. And then the diameter of the plate is no wider than 150 millimeters in diameter, meaning we always use the same size of a plate. You use a non-toxic swab to spread the bacteria on the agar plate to obtain confluent growth. And that's why I said the bacteria grows everywhere, and we call it a lawn of bacteria. In this lab, we'll see how you do that. You then place the disc at least 24 millimeters apart and no more than 12 discs on the plate. So right there, no more than 12 discs, and all the discs have to be at least 24 millimeters apart. They can be further, but they have to be at least 24 millimeters apart. Where were we? Then we add the antibiotic disc 5 minutes, but no longer than 15 minutes after you add the agar inoculation containing the bacteria. So what you're doing is you're allowing the liquid that you put on the plate to be absorbed by the agar for 5 minutes before we put on the antibiotic disc. But we make sure we add the antibiotic disc not longer than 15 minutes after adding the bacteria. We then incubate in an incubator no longer than 15 minutes after adding the antibiotic disc. And we always incubate in a non-CO2 incubator for total hours of 16 to 18 hours at 35 degrees Celsius. And we do all of this to control the variables so that when one person does the Kirby-Bayer distaffusion, they will have a similar zone of inhibition to somebody else in another part of the world who does the same Kirby-Bayer test, and their zone of inhibition will be similar. And if we didn't do the standard procedure, that size of zone of inhibition could differ, and it could differ significantly. Any question about any of that? All right, after incubation, the inhibition zone size is then measured using a ruler that has millimeters and then you record the size in millimeters. You then look up in the chart, and it's a chart made by the National Committee for Clinical Laboratory Studies that sets the standards for interpreting the zone of the size where the bacteria is considered to be sensitive to the antibiotic, or the bacteria is considered to be resistant to that particular antibiotic. And you'll note that the bacteria could be intermediate, meaning if the size of the zone of inhibition is smaller than a certain size, it's not sensitive, but it is smaller than the size of the resistant colony, then we call it intermediate. And let's get an example here. Here we have a disc and the zone of inhibition on a plate. And we can see that this is a zone of inhibition of 20 millimeters. And here's the table. This is the antibiotic penicillin. Each antibiotic has a different table, and each organism it's applied to can have a different table. And this is Staphylococcus aureus that's been put on the agar plate. And when we see here on the standards from the NCCCLS, that if the organism has a zone of inhibition of 20 millimeters or less, we say it is resistant. And since this is 20 millimeters, this species, Staphylococcus aureus, is resistant to penicillin, and that's probably penicillin G. But if the size of the zone of inhibition were be between 21 and 28 millimeters, then we'd say it was intermediate. The antibiotic was intermediate in its effectiveness on this Staphylococcus aureus. And if the size of the zone of inhibition was greater than or equal to 29 millimeters, we'd say the organism Staphylococcus aureus was sensitive to this antibiotic. Any question about any of that? So that's how you determine whether an antibiotic is sensitive or resistant. You actually have to measure the size of the zone of inhibition. You take the diameter, and usually you measure it two different ways. You measure it on this angle and that angle. This one looks circular, but sometimes they're not quite circular. And then you take the average, and if the size is smaller than the resistant, then we say that this antibiotic, the bacteria is resistant to that antibiotic. If it's larger than the size for sensitive, you say it is sensitive to the antibiotic. And if it is somewhere in between, you say it's intermediate. Any question about any of that? All right. So here's the terms. You should know the terms for this lab. Go through that. You don't need to go through the references. In this laboratory, you're not physically going to be performing any of these activities. Everything's going to be done online. You will be responsible for knowing how to interpret the effectiveness of an antibiotic using the Kirby-Bauer disc diffusion test. In procedure 1, you're going to be preparing a spread plate, and we have a little video on how to prepare a spread plate. Review the steps using aseptic technique when preparing a spread plate. And this will make a bacterial lawn of growth of bacteria. What you do is you put the bacteria on a cotton swab, remove the excess liquid from the cotton swab by touching the side of the sterile part of the inside of the tube, and then inoculate as much of the agar surface as you can with the swab. Let me try and demonstrate that. Can you guys see this? We're going to pretend this is the Petri dish, and this is my cotton swab. It has the bacteria on it. Can you guys see this? Nobody's saying anything.
Yes.
Okay. So you swab the plate everywhere. So everywhere it gets covered. Then you rotate the plate a fourth of the turn and rotate the swab a fourth of the turn. I don't know if you saw it with the pen, but, and then you do the plate one more time, pretend it's round, where you inoculate the bacteria everywhere. And then you turn the swab a fourth of the turn, turn the plate a fourth of the turn, and then swab again everywhere. I'm going to quickly do it. And then you do it a fourth time. Sometimes people only do it 3 times. This one says only do it 3 times. Swab it everywhere. And then at the very end, come around the edge of the plate with the swab and cover the edge of the plate. The reason is when you're going up to the edge, you may not quite get everything. So we go around the edge to make sure the bacteria is growing everywhere on the plate. Then you put the cotton tip swab in the biocide and then allow the plate to sit undisturbed to absorb that liquid into the agar. Then you apply the antibiotic disc on the plate. And then you incubate the plate upside down in the incubator overnight for 16 to 18 hours at 35 degrees Celsius. You then look at the zone of inhibition and then measure the diameter of the zone of inhibition. Okay. So for procedure 2, we're evaluating the effectiveness of an antibiotic. We have a little video on preparing a culture for the Kirby-Bayer dysdiffusion. So watch that. And then we have another video on interpreting the results for the Kirby-Bayer dysdiffusion. So watch that. And then do the link on the virtual lab. And I've got that open already. Well, it used to be here. It has to refresh. Okay. So you can read about it if you want. You can also skip that and just click start. And then it kind of tells you what to do. Click the menu button. There's the ruler. And it's got the different table of what something is sensitive. And if the zone of inhibition for amoxicillin, AMC, does anybody see that? Right there. If it's greater than or equal to 18 millimeters, then we say it's sensitive. And we can look at that right there. Amoxicillin is very small, so it's obviously not sensitive. And it looks like it's less than 13 millimeters. Let's bring that over here just to make certain. Well, it's about 12 millimeters, but it's still less than 13. So we say that this bacteria is resistant to amoxicillin, and the bacteria is right there, it's E. coli. Any question about any of that? So click the menu and it will tell you how to go about doing this. And when you did restart, it gives you the different bacteria. And this one there, CIP, you bring the ruler up and then measure the size of the zone of inhibition. That looks like it's very close to 4 centimeters, which would be 40 millimeters. And so CIP would be greater than 17 millimeters. So we'd say that one is sensitive. Any question about any of that? How you work the virtual lab? Now, how do you go through the different steps here? There we go. Measure, it tells you how to do it. Hit the steps, take the virtual ruler, measure the zone of inhibition, compare it to the table, and then record the data as R, I, or S. All right, I'm not seeing how you get down and start recording. Oh, here we go. You click on the table here for what it is. And if it's green, that's your selection. Let's just go VA. I'm not going to measure it, but I'm going to say it's sensitive. And how do I get the results? Someplace it gives you what the answers are. Well, hopefully you'll figure it out because I'm not doing it quickly. There we go. If it's green, it's the correct result. If an X, you select that, that means it's the incorrect result. That's the incorrect result, so that has to be the correct result. All right, so that's how you tell whether it's correct or not. If it's green, it is correct. If it's an X, it means it's incorrect. I knew there's a way. All right, any questions on how to do the virtual lab? It will take a little bit of time to load, so give it time to load, especially if you have a slow internet connection. And then using the standards required for the Kirby-Bayer susceptibility test, using E. coli and the prepared spread plate technique, we're going to look at the antibiotics disc test placed on an agar surface and incubated overnight for 35 degrees Celsius. Below are the results with the NCCLS zone standards used for interpretation. And we have calipers here. And then there's the table. Remember, we're only doing E. coli, so this part of the table here, not Staphylococcus aureus. And what you do is you measure the zone of inhibition using the calipers. And I'll warn you, you can't read from this part of the caliper to that part because this scale is not over here, it's starting there. So how you read this is you look where this 0 is, and we're going to read ampicillin as being 15 millimeters in diameter. And then come down to the chart. So I'll do the first one for you. Ampicillin is 15 millimeters, and that is E. coli, correct? Let me make sure. E. coli, yes. So that would be intermediate for ampicillin. All right, and so you put in here the size of the zone of inhibition and put in an I. And the table will allow you to type in it. Okay. All right, so finish the table and then work on question 3. List the variables that must be standardized when performing an antibiotic susceptibility test using the Kirby-Bayer disc diffusion method. And you have to fill out each of the, I don't know, asterisks or whatever that's called. 1, 2, 3, 4, 5 different steps or items. All right, question 1, give the size of the zone of inhibition. Often varies for 2 different species of bacteria when challenged with the same antibiotic. What's a possible explanation for these differences? 2, what would happen if we didn't use aseptic technique to put the antibiotic disc on the plate? And I guess we should say, you should explain that, meaning you're going to get contamination. I know that. What will the contamination do to the results of the antibiotic disc? And 3, what would happen if we increase or decrease the concentration of the antibiotic in the disc? You do have to explain it in both increasing and decreasing the concentration of the antibiotic. Antibiotics can be bactericidal, meaning they kill the microbe, or bacteriostatic. They inhibit microbial growth, but do not kill the bacteria. When you're observing a size of the zone of inhibition around a disc, how would you determine whether the antibiotic was bactericidal or bacteriostatic? You have to think about this one. Okay? Meaning this one often students get wrong because it isn't mentioned in the lab manual. And I didn't mention it in the presentation, although I sort of hinted at it. When performing antibiotic sensitivity testing, it is important that the test conditions are kept very standardized, meaning you use same kind of media, you have the same agar depth, the same age of the culture, you're applying the same number of cells. Why do we keep it so standardized? All right, 6, are there any other substances besides antibiotics that we could use to evaluate using this method? And this one is really easy to answer. 7, does the presence of the zone of inhibition necessarily mean that the bacterium being tested is susceptible to the antibiotic? So you have a paper disc. I guess we'll go right there. We have a zone of inhibition. Does that mean that the bacteria is sensitive to that antibiotic? All right, that's it. Any questions? If not, go ahead and get started on the lab. And I will be here until 8 o'clock to help answer any questions. I may step away from my desk, but I'll be back in just a few minutes if you have a question. All right, let's start resuming. All right, there was a question asked, and you're saying the worksheet doesn't have a question that the lab manual does?
Yeah, so the worksheet to lab 6 starts on procedure 2, question 2. It doesn't have any procedure 1 on it, or the instructions or links for procedure 2, 1, which is fine.
Let me get the correct lab manual because I'm using the one I talk about.
Yeah, the manual has everything on it. It just doesn't match the worksheet.
Well, follow the worksheet generally. Let me take a look at what's going on here. Lab 6. It's just that I've got the older version of the lab manual because I've got highlighted what I want to talk about. So what question are you referring to is in the lab manual? It's not in the worksheet?
So in the lab manual, it says laboratory 6 exercises, procedure 1, preparing a spread plate. That entire procedure is not on the worksheet for lab 6, but it looks like the only actual question it's asking is number 3, using words explain.
Procedure 1. Which question? I don't see a question here. Oh, you're not seeing the screen. Just a minute. All right, so here's procedure 1. What question are you referring to?
So write down there question 3, using words explain the spreading pattern that you would use to obtain confluent bacterial growth on and on.
You don't need to answer that. It's discussed in here and that it should go everywhere. All right, so that's not a question. It doesn't have a question. But yeah, I understand. That's not on the worksheet. You don't need to answer it. Is there anything else that's different?
Nope, it's starting from procedure 2, question 2. All the questions are on there, so.
Okay.
Thank you.
All right.